So good morning, everyone, and thank you very much for this invitation. Today we'll start the emerging pollutants session talking about this type of chemicals on coastal aquifers. So first of all, this work I've done have been recently published in Journal of Hazardous Materials. So I will describe the paper we did and all the results we have during this project. So first of all, what are these emerging pollutants? So there are a wide variety of chemicals that we call emerging pollutants, and all of these chemicals may enter in the environment. Once they enter, they can have a potential hazardous effect into this environment, and usually they have not a regulation because they are quite new, or maybe because regulation is, let's say, late for them. So one example about emerging pollutants is BPA, bisphenol A. Maybe if you have a tupper, when you have lunch, a plastic tupper, you probably will see that is BPA free. Well, it could be true, but there are new bisphenols, for example, that have been created later, such as bisphenol S, bisphenol F, or bisphenol AF, that are very similar structurally to bisphenol I, and we know nowadays that they are more or less equally hazardous effect. They have the same hazardous effect or even more. So yes, we are changing the bisphenols, but as example, but we are not saving the planet, let's say, in this sense. So I also need to give some information about coastal aquifers. In all coastal aquifer, which is an aquifer between the land and the ocean, we have two complementary processes. The first one is the intrusion, which is the water from the sea going through the aquifer. Oh, now it's better, right? Okay. Okay. So we have the water going from the sea to the aquifer, but also we have the discharge. I'm going to talk today more about the discharge, which is the water from the aquifer through the sea. Okay, which is our hypothesis? So we consider that analyzing emerging pollutants, we can monitor and clarify the effect of the discharge. We can better understand the discharge by knowing which emerging pollutants we have in the aquifer. And we will use liquid chromatography coupled with high-resolution mass spectrometry, which is an analytical chemistry tool that I am used to use. So, which are our two objectives? The first one is, let's say, the easiest one to achieve, is to know which chemicals, which emerging pollutants we can find along the aquifer. And the second one, maybe the riskier one, is not only knowing which chemicals and also the distribution, also evaluating if these chemicals may help us to understand the discharge, how this water goes from the aquifer, from the land, through the sea. So for that, we have an experimental site that we have been working in since 2014, in which we have boreholes that are in the same longitudinal way, and all of them are, we can take samples at different depths. So we have this distribution that we can see in the picture. So what we do? We take 4 liters of each sampling point, we filter all of them, we pre-concentrate them to have a better picture of the chemicals inside, and we analyze all of them in a Q-exactive, in an orbit trap. So we pass from 4 liters to 4 milliliters in total, so it was a quite high pre-concentration. And we perform a target screening. Which is this target screening? So when we want to analyze emerging pollutants, for example, we can look for known chemicals. So we can buy standards, we can analyze them, we will have a very good and very reliable information about these emerging pollutants, but we are only going to see these emerging pollutants that we bought the standard, that we previously selected. If we don't select an analytical standard, we don't buy them, so we are not going to see them, even if it's in the aquifer. So even if they're a very good method, they have this limitation. The Iceberg picture is quite good for that. We are going to see only those chemicals that we look for them, but most of the potential stressors are not going to be analyzed, so they are going to be omitted, and they may have this hazardous effect. And we also need to pay for this standard, so we need more methods, we need more alternatives. So that's why we started to use non-target screening. Thanks to this high-resolution mass spectrometry, we can work with this type of tool that has been developed in the last year, so it's more used now because of the evolution of the instruments. And now we can look for unknown substance. What does I mean? This means that we don't need to buy the standard, we don't need to pre-select the chemical before the analysis. We can do the analysis, and once we have done the analysis, we can look for all these chemicals. So it seems to be much better, but we need to be clear that we need acquisition data tools much more complicated. We will have probably thousands and hundreds of features, that are like chemicals, but we don't know which chemical is it feature. So we need to spend many time to prioritize which chemicals we really want to analyze, to identify, and go for them. So it's maybe the tools that we have later are more time consuming. So what we did, we used both the target and also the non-target screening, so we could have a better picture of the aquifer. And as our first objective was to know which chemicals and the distribution of these chemicals were in the aquifer, we found 92 chemicals. We have biocides, pharmaceuticals, we also found food-related chemicals like caffeine. There was some cocaine also in the aquifer in some samples, so it was quite contaminated, the aquifer. Also personal care product that maybe people can use daily and they finish in the environment. But we only, apart from these 92 chemicals we found, we had this second objective, not only knowing which chemicals, also see if we can understand better the discharge. So for that, we prioritize. From these 92 chemicals, we only keep those that were in a high part of the samples, more than 75%, and also those which correlate with salinity, because salinity helps us to understand if there is intrusion and discharge, because the different salinity between the fresh water, brackish water, the water which is mixed, and the saline water. So after doing that, we found that 8 chemicals correlate with this salinity. 6 of them were PFAS, perfluorinated chemicals. We will have more information about them today. And this type of chemicals, as well as the pharmaceuticals, topiramate and carbamazepine, all of them are very recalcitrant. They are very persistent in the environment. So this is why they didn't degrade along the aquifer conditions, and they stay along the system. So I want to especially focus on PFAS, because they are called the forever chemicals. Their degradation is expected to be very difficult, and as we can see in the picture, the size of the ball means the concentration we find in the aquifer. When we go to the shallowest part of the aquifer and to the far away from the sea, we can see higher balls, bigger balls, which means higher concentration. While we go deeper, we see that there is this dilution of the chemical. We see less compound. But we also did this non-target screening. As a reminder, we have more features, but we need to prioritize and identify those chemicals that we are interested on. So after having more than 5,000 features, we focus on those which were in a high percentage of the sample, and also those which correlate with salinity, the same process that we follow with the target screening. And only 3 features accomplish these 2 requirements. What happened with the 2 first one? Now we don't have we have the mass of the chemical, but we don't know which chemical. After working on it, we find out that it was another PFAS. The main one of them was the other PFAS, and also a fragment of this PFAS. So what does it mean? That at the end we found, with the non-target screening, another PFAS different that hasn't been reported in the environment yet in our samples also. So as a conclusion, to accomplish our first goal, we found 92 chemicals in the aquifer with this target screening. All of them were from anthropogenic origin. Maybe if we use pesticides to apply to the crop, they can end up in the aquifer, for example. And we know this distribution because of the sampling campaign. And also, about the second target of our analysis, we found 7 PFAS as a main result that were in the aquifer by the 2 methods, both the target and also the non-target screening, which correlate with this salinity and which end up also in the sea. So after this, I would like to acknowledge to all the authors and co-authors of the paper, of the project, and also, of course, to the financiation that we have to carry out all these experiments. And thank you very much for your attention. If you have any question.